You are standing at the edge of a wild grass field. It is roughly 8,000 years ago, somewhere south of the Caspian Sea. The air is dry, the sun is high, and the plants moving in the wind around you look like nothing that could change human history. There is no harvest underway, no fire, no grinding stone, no farmer walking between the stalks. There is no human being anywhere nearby at all. There are only two plants growing close enough together that their pollen occasionally drifts from one to the other. One is a kind of wheat. Early farmers living somewhere in the wider region already gather it, protect it, and plant its seeds. It is useful, familiar, already halfway between a wild grass and something that might eventually become a proper crop. The other plant is a weed, a wild goatgrass. It is tough, thin, and thoroughly unremarkable. It produces almost nothing an early farmer would have wanted. Most people working these fields would have pulled it from the ground and thrown it aside without giving it another thought. Neither plant looks extraordinary on its own. And then, in a single growing season, something happens between them that should almost certainly fail. Their pollen combines. Their chromosomes, which have no reason to fit together properly, somehow fuse. In almost every case, a hybrid formed between two different plant species would be sterile. It might grow once, but it would never produce fertile seeds of its own. The experiment would end before it truly began. This time, it does not. Through an exceptionally rare chromosomal accident, the new seed becomes fertile. It sprouts, it grows, and it produces another generation. Nobody plants it deliberately. Nobody selects it. Nobody is even watching when the event takes place. There is no ancient farmer carefully crossing two plants together. No breeder trying to improve the quality of flour. No plan to create stronger dough, softer bread, or a taller loaf. There is only a crop, a weed, and an accident. But the new plant carries genetic material from both parents. And part of that genetic inheritance will eventually help wheat dough stretch around trapped gas instead of tearing apart. It will help dough rise higher, hold its structure, and produce the soft, open texture associated with much of the bread people eat today. That unnoticed plant growing at the edge of an ancient field becomes part of the direct ancestry of modern bread wheat, formerly known as Triticum aestivum. Its descendants will eventually be planted across continents, milled into flour, mixed with water, and baked into everything from supermarket sandwich loaves to village bread pulled from a clay oven. And the humans who will spend the next 8,000 years building farms, cities, trade networks, religions, governments, and entire civilizations around that grain have absolutely nothing to do with creating the genetic accident that made it possible. So, how did humans invent bread? The honest answer is stranger than it first appears. Because humans invented the tools. They developed the techniques, they learned how to harvest grain, grind it into flour, mix it with water, preserve old dough, control fermentation, and apply heat. But some of the most important parts of a risen loaf were never invented by humans at all. If tracing an invention back through the parts nobody actually invented is the kind of investigation that keeps you coming back, subscribing means the next one finds you without you having to look for it. One was created by an accidental collision between plants. The other was alive, invisible, and misunderstood for thousands of years. To understand bread, we have to begin with wheat. And wheat is far more complicated than it looks. A grain of modern bread wheat appears simple. It is small, dry, and ordinary. But genetically, it is one of the most complicated major crops humans have ever depended on. Most animals and many plants carry two sets of chromosomes, one set inherited from each parent. Bread wheat carries 6. Geneticists call it a hexaploid. Those 6 sets are grouped into 3 related genomes, usually called A, B, and D. You can think of bread wheat not as one plant with one simple family history, but as a biological archive containing the combined inheritance of several different ancestral grasses. And it did not acquire those genomes in one step. The first major event happened before modern bread wheat existed. 2 wild grass relatives crossed and formed a new wheat lineage with 4 sets of chromosomes. This tetraploid wheat eventually included emmer, one of the important grains cultivated by early farming communities across the Fertile Crescent. Emmer could be harvested, ground, cooked, and used for bread. It was not useless or primitive. For thousands of years, it was a major human food. but it did not yet contain the complete genetic combination found in common bread wheat today. Then came the second event. Cultivated emmer wheat grew close to a wild goat grass called Aegilops tauschii. The goat grass was not a valuable crop. It produced small seeds and had little reason to attract the attention of early farmers. Yet it carried something that emmer did not, the D genome. When emmer and Aegilops tauschii crossed, the resulting plant inherited genetic material from both. Through chromosome doubling, the hybrid became fertile instead of reaching the usual sterile dead end. The new wheat now carried 6 sets of chromosomes, 2 from each of 3 ancestral genomes. This was the biological foundation of bread wheat. It is tempting to imagine a single field, a single plant, and a single miraculous seed from which every later wheat plant descended. The real history may not be that clean. Genetic evidence cannot give us the exact day, the exact field, or the exact farmer who first noticed the new grain. Researchers continue to debate whether the crucial hybridization happened once or more than once across a wider region where cultivated wheat and wild goat grass overlapped. The date is also an estimate. Genetic dating works by comparing modern genomes, measuring accumulated differences, and calculating backward. It can provide a probable window, not a calendar entry. So we should be careful. We cannot say that one specific plant appeared on one known morning 8,000 years ago. We can say that sometime during the early history of agriculture, cultivated tetraploid wheat crossed with Aegilops tauschii and produced the hexaploid lineage that became modern bread wheat. And that new genetic combination changed what dough could do. When flour is mixed with water, proteins inside the grain begin interacting. 2 broad protein groups, glutenins and gliadins, contribute to the network we call gluten. Gliadins help dough stretch. Glutenins help it resist and return. Together, they create a structure that can expand without immediately tearing apart. The D genome did not single-handedly invent gluten. Wheat's baking properties come from genes distributed across its combined genomes. But the contribution from Aegilops tauschii included important variants that improved dough strength and bread-making quality. This matters because fermentation produces gas. If a dough cannot hold that gas, the bubbles escape. The dough may still become food. It may still become a flatbread, a dense loaf, or a cooked paste, but it will not rise in the same way as a strong wheat dough that stretches around thousands of tiny pockets of carbon dioxide. This is why the wild goat grass matters. It did not create bread by itself. Humans had already been processing grains long before common bread wheat appeared. People could make flatbreads from wild cereals and other starch-rich plants. But the new wheat gave bakers a grain especially suited to tall, airy, fermented loaves. And the strangest part is that early farmers did not know any of this. They could not see genomes, they did not understand chromosome doubling, they did not know why one wheat behaved differently from another. They simply noticed useful results. Perhaps the new wheat produced seeds that were easier to use. Perhaps it performed well in local soil. Perhaps its flour made dough that was more flexible or more reliable. Whatever advantages people saw, they saved seeds from plants that worked and planted them again. Invention is often described as a moment when a person understands a problem and deliberately designs a solution. Bread was not like that. No single person understood the entire system. No single generation created every part. Bread emerged from a chain of partial knowledge. Someone learned that hard seeds became easier to eat when crushed. Someone learned that flour and water could form a dough. Someone learned that dough cooked on a hot surface became portable food. Someone noticed that old dough could make new dough rise. Someone saved seed from wheat that behaved better. Each person understood only the part directly in front of them. Behind those visible actions were invisible systems that no one could explain. The first was genetics. The second was fermentation. Imagine an ancient baker preparing dough. There is flour in a bowl, water, perhaps salt, and a small piece of yesterday's dough. That old piece smells sharp. Its surface may be cracked or slightly wet. It does not look like a machine. It does not look alive. But when it is mixed into fresh flour and water, the dough begins to change. It softens. It expands. Bubbles form inside it. The smell becomes more complex. If the baker waits the right amount of time and then applies heat, the result is lighter and more flavorful than a simple mixture of flour and water. The baker does not know why. For most of human history, nobody did. People learned to preserve starter cultures long before they understood that those starters contained living communities of yeast and bacteria. They fed them, divided them, shared them, and passed them between households. A starter could survive longer than the person caring for it. It could move with a family from one settlement to another. It could become part of a local tradition. But to the people using it, fermentation was not microbiology. It was behavior. Feed the starter. Keep it warm. Do not let it dry out. Use part of it. Save the rest. Repeat tomorrow. The organism most closely associated with bread fermentation is Saccharomyces cerevisiae, the same species widely used in beer and wine. Yeast consumes available sugars and releases carbon dioxide and alcohol. In bread dough, the carbon dioxide becomes trapped inside the gluten network. In the oven, the gas expands, water becomes steam, proteins set, starches gelatinize, and the temporary foam becomes a permanent crumb. The alcohol mostly evaporates. The yeast dies. The bubbles remain. That is what rising bread is, the preserved shape of microbial metabolism. Yet, until the 19th century, bakers could not see the organisms responsible. They could observe effects, but not causes. For centuries, fermentation was explained through vague ideas about decay, chemical transformation, or spontaneous processes within the material itself. The decisive change came in the work of Louis Pasteur. In 1857, Pasteur demonstrated that fermentation was driven by living microorganisms. His research was connected to practical problems in wine and vinegar production. Producers wanted to know why some batches fermented correctly while others spoiled. By examining fermenting liquids and identifying different microorganisms associated with different outcomes, Pasteur helped establish that fermentation was biological, not merely an abstract chemical change. This discovery transformed the meaning of something bakers had been doing for thousands of years. The bubbling starter was not simply active flour. It was an ecosystem. The dough did not rise because old dough carried a mysterious force. It rose because living cells were feeding, reproducing, and releasing gas. Think about the gap between use and understanding. For thousands of years, human hands touched dough every day. Bakers watched it swell. They smelled it. They adjusted temperature and timing. They knew when it was weak, when it was ready, and when it had gone too far. They developed skill without explanation. A baker could know more than a scientist about how a starter behaved while knowing nothing about the microorganisms inside it. This was not ignorance in the simple sense. It was a different form of knowledge, repeated observation, memory, and practice refined across generations. Modern science has made the invisible visible, but it has also shown that fermentation is more complicated than a single yeast species. Traditional sourdough starters contain communities of yeasts and lactic acid bacteria. These organisms compete, cooperate, produce acids, change flavor, and alter the conditions inside the starter. A healthy starter is not one organism living alone. It is a small, changing ecosystem. That ecosystem is shaped by how it is treated. A popular story says that sourdough flavor mainly comes from wild yeast floating in the air of a particular city. According to this idea, a starter made in one place should have a microbial identity determined by local geography. The reality appears more complicated. Large studies of sourdough starters have found that location alone explains only a limited amount of the variation between microbial communities. The flour used, the feeding schedule, temperature, hydration, storage conditions, and the habits of the baker can all influence which organisms become dominant. Two starters kept far apart may become similar if they are fed in similar ways. Two starters in the same neighborhood may become different if their owners use different flour and routines. This changes the way we think about the baker's role. A baker is not simply capturing a local organism and preserving it unchanged. The baker is creating an environment. Every feeding changes that environment. The choice of flour changes the available nutrients. The amount of water changes movement and acidity. Temperature changes which organisms grow faster. Time between feedings changes who survives hunger. Households that kept starters were performing repeated selection. If one culture made good bread, people saved it. If another smelled unpleasant, failed to rise, or spoiled, people discarded it. Useful microbial communities were more likely to continue. Unsuccessful ones disappeared. This is a kind of domestication. Not a planned domestication based on genes or laboratory measurements, but a slow filtering process based on results. Rise well, taste good, survive until tomorrow. Those were the selection rules. Over many generations, yeast strains associated with human fermentation developed genetic patterns different from wild populations found on tree bark, fruit, and other natural environments. The history is not one simple line from one wild ancestor to one domesticated baking strain. Yeast populations have mixed, separated, and adapted in complex ways. But the larger pattern is clear. Long association with human fermentation changed them. Humans were selecting microorganisms before they knew microorganisms existed. Bread, therefore, sits at the meeting point of two forms of unconscious breeding. Farmers selected wheat plants by saving useful seeds. Bakers selected microbial communities by saving useful starters. Neither group needed to understand genes. Neither needed microscopes. Success itself was the guide. But the wheat story and the yeast story are not identical. The crucial hexaploid wheat lineage began through a rare natural hybridization that humans did not plan. Once it appeared, people preserved and spread it through agriculture. Fermentation organisms were already present in the environment. Humans repeatedly captured, maintained, and favored communities that produced desirable results. In one case, nature created a useful genetic combination, and people kept it. In the other, nature supplied invisible organisms and people built an environment in which useful ones could survive. Bread required both. The flour needed physical properties capable of forming a stable dough. The microorganisms needed sugars to consume. The dough needed time. The baker needed experience. The fire needed control. Remove any one part, and the result changes. This is why asking who invented bread is more difficult than it sounds. We often want inventions to have names and dates. We want a first baker, a first loaf, a first moment of discovery. But bread is older than written records, and its development was distributed across many communities. Flatbreads could be made from grains long before the appearance of modern bread wheat. People had already learned to grind wild plants, mix them with water, and cook them. Those early breads did not depend on the exact genetics of today's common wheat. So the genetic accident that produced bread wheat did not invent all bread. What it did was contribute to a grain that became especially important for risen bread. That difference makes the story more accurate and, in some ways, more interesting. A baker in an ancient village could control fermentation without understanding yeast. A farmer could improve wheat without understanding chromosomes. A modern baker may understand neither genetics nor microbiology in detail and still produce excellent bread because the practical system has been carried forward. Knowledge can live in actions. A recipe is stored behavior. A starter is stored ecology. A seed is stored evolution. Put them together and you have bread. The story continues in modern laboratories. Aegilops tauschii, the wild goatgrass that contributed the D genome to bread wheat, still grows across parts of Iran, the Caucasus, and Central Asia. It remains useful not because it gives us a romantic connection to the past, but because it contains genetic diversity that modern wheat may have lost. Domestication and selective breeding produce advantages, but they also create bottlenecks. Farmers repeatedly choose a limited number of plants with desired traits. Over time, successful varieties spread widely, while other genetic possibilities disappear from cultivated fields. Modern bread wheat is productive and useful, but its genetic diversity represents only part of what existed in its wild relatives. That matters in a changing climate. Researchers search wild Aegilops tauschii populations for genes connected to drought tolerance, heat tolerance, and disease resistance. Traits ignored by ancient farmers may become essential under modern conditions. A weed once considered unimportant may contain biological tools needed to protect future harvests. But moving those traits into commercial wheat is not simple. Modern breeders sometimes recreate a version of the ancient hybridization deliberately. They cross a tetraploid wheat, often durum or a related line, with selected Aegilops tauschii plants. The result is synthetic hexaploid wheat. In effect, scientists repeat the old accident on purpose. This time, they know the parent plants. They can examine the chromosomes. They can track genes. They can test resistance, yield, and quality. What happened unknowingly in ancient fields can now be reconstructed under controlled conditions. That is one of the deepest ironies in the story. Modern science is using advanced genomic tools to recreate a process nature performed without intention thousands of years ago. The same thing is happening with sourdough. Microbiologists can sequence the organisms inside a starter and identify which yeasts and bacteria are present. They can connect particular microbial communities with acidity, aroma, fermentation speed, and texture. They can investigate why some organisms repeatedly appear together and why others exclude one another. Bread looks simple because the difficult work happened before the ingredients reached the bowl. Inside the flour is a history of hybridization, chromosome doubling, mutation, selection, migration, and agriculture. Inside the starter is a community shaped by thousands of feedings, countless kitchens, changing temperatures, different flours, and generations of selection. Inside the recipe is human memory. The person making bread today does not need to repeat the original discoveries. They inherit them. Every time someone feeds a starter, they continue a practice developed before microorganisms had names. Every time someone kneads wheat dough, they use properties created by ancient plant evolution and preserved by farming. Every time a loaf rises, invisible cells fill an elastic protein network with gas. And every time heat sets that structure, a temporary biological process becomes food. So, how did humans invent bread? They did it slowly. They did it without understanding most of what was happening. They crushed seeds because crushed seeds were easier to eat. They mixed flour with water because the mixture could be shaped. They cooked it because heat transformed it. They saved the wheat that worked. They saved the dough that rose. They repeated successful results until accident became tradition. But humans were never working alone. A wild goatgrass contributed genetic material that strengthened the future of wheat dough. Microorganisms entered flour and water, consumed sugars, and filled that dough with gas. Plants, fungi, bacteria, fire, weather, and human habit all became part of the same system. Bread was not invented in a single flash of intelligence. It was assembled across thousands of years through cooperation between human observation and processes humans could neither see nor explain. That does not make bread less of a human achievement. It makes the achievement more honest. Human intelligence is not only the power to design something from nothing, it is also the power to notice, to remember, to repeat, to care for a process before understanding it, to keep a useful seed, to save a piece of old dough, to learn from results and pass that learning forward. The first bakers did not understand genomes. They did not know yeast was alive. They did not know that the bubbles inside dough were carbon dioxide. They did not know that a weed growing beside a field had contributed part of the genetic structure inside their grain. They only knew that some dough rose better, some bread tasted better, some methods were worth repeating, and they repeated them. That repetition became knowledge. The knowledge became tradition. The tradition became civilization's most familiar food. The next time you hold a loaf of bread, look at it closely. The crust is the result of heat. The crumb is the fossil of trapped gas. The flavor is the work of chemistry and living cells. The flour carries the inheritance of several ancient grasses. And the loaf itself carries the memory of people who learned to work with all of these things long before they could explain any of them. Bread looks like a human invention, but inside it is a genetic accident nobody planned and a living partnership nobody could see. Humans did not create every part. They did something more patient. They noticed what worked. They protected it. They repeated it. And over thousands of years, they turned accident, biology, and habit into bread.